I'm going to talk about mycorrhizal fungi. I started working with them. I hate to say because it ages me, but 1979 when I was at Utah State, and then I did a master's on mycorrhizal fungi, jump starting uh, succession on disturbed sites like mine sites and that kind of stuff. And then I went and I worked on a National Science Foundation grant monitoring mycorrhizal fungi and fungi on Mount St. Helens after the volcano blew. And then I went to Oregon State, did a PhD working on mycorrhizal fungi and interactions with plants. And then did a postdoc also working with mycorrhizae and more interactions with different soil organisms that benefit plant growth. And I went from there to Mexico and worked for their version of the National Science Foundation in Mexico, looking at how we're looking at how mycorrhizal fungi allow plants to communicate back and forth. And that's kind of what I did down there. And then I came back up here and wound up going to with ARS and the Plant Gene Bank, and then with NRCS. Well, part of it with Palmer Soil and Water, and then part of it with NRCS. But anyway, so we're going to look at mycorrhizal fungi, which are keystone species for soil health. And in my opinion, anybody that this is strictly my opinion, but anybody who talks about soil health without using mycorrhizal fungi in the same sentence is barking up the wrong tree. Mycorrhizal fungi are keystone species, which is a plant or an animal or microbe that has a large impact on its ecosystem relative to its abundance. So their importance is much greater than the sum of their numbers. Okay, and keystone species are vital for the long-term health of an ecosystem. I'm also going to talk a little bit about keystone mutualists, which are two or more species of organisms that engage in reciprocity and reciprocally vital interactions. So the disruption of one of these species, let's say it's a plant, or let's say it's a mycorrhizae, impacts the other, and ultimately the ecosystem as a whole. These pairs are often like pollinators, so like hummingbirds that rely on specific plants for sustenance, and that rely on and the plants rely on those pollinators to reproduce. And here's a picture of Scutalospora, which just means it's an arbuscular mycorrhizae, an endomycorrhizae, and I'll talk about that. But it is this is from a cardone cactus down in Mexico. And then the two upper right-hand pictures are winter wheat, and that's mycorrhizal hyphae encircling winter wheat. And then the bottom right-hand side is an arbuscule. And the endomycorrhizae, the largest group of endomycorrhizae are the arbuscular mycorrhizae. An arbuscule just means tree-like, and you can see how that organ or that fungal structure there looks a lot like a tree. The really interesting thing is that endomycorrhizae and arbuscular mycorrhizae will penetrate the cell wall, but not the cell membrane. I don't know if you guys know that, but human beings have a cell membrane. That's all we have. But plants have a cell wall, a rigid cell wall, and a very plastic, which means it's malleable, a very plastic cell membrane on the inside of that cell wall. So if you could imagine what this structure looks like, it's like, imagine, I was trying to make one of these, but imagine gluing a rubber glove to a hole that you put in a box, and then reaching your hand in through the hole into that plastic glove or that latex glove. That'd be the fungal structure, and the latex glove would be the plant membrane. My outline is going to be, let's see, just exactly what are these associations? And then I'm going to look at some benefits and disadvantages of the association, what plants respond and which ones don't. There's a lot of misinformation out there. And then I'm going to talk about 7 different types of associations. That's another problem that I see that people have. They do not realize there's 7 different types of mycorrhizal associations. And then, thanks to Rita Joe, I gave a talk. I gave a talk up in Fairbanks in 2013, and I got back home from that talk, and Rita Joe convinced me to do a flow chart that kind of helps people to walk through what can be a kind of a complex problem or complex association. So what are these associations? Of course, mycorrhiza or mycorrhizae, which is plural, literally means fungus root or fungi root. And when you see the term mycorrhiza or mycorrhizae, that is not in reference to the fungus. That is in reference to the association. And I have an issue with this because ProMix puts mycorrhizae on their ProMix bales, and that means that they've got literally a living root or a living plant inside that bale. That because they're mislabeling what they're doing. Okay. So how long have mycorrhizal associations been around? About 460 million years. So they got a bit of a head start on us. In fact, they're responsible for allowing plants to colonize dry land. Mycorrhizal fungi are in 80 to 90% of our plant families. So, in essence, in nature, this mycorrhiza association is the rule and not the exception. So, there are places you can go where 90 to 99% of the plant cover that you look at is all mycorrhizal. And the unique thing about mycorrhizal fungi, and this is why I talk about soil health and the association of a plant and its the soil around it, is that mycorrhizal fungi are the only organism that occurs both inside and outside of the host plant at the same time. Taking a drink of water. Okay, when you get plants, when plants become mycorrhizal, they totally change what a plant exudes out into the soil. So you no longer have uh, what is called a rhizosphere, which is that a rhizosphere is that 2 millimeters of soil around the root. Now you have a mycorrhizosphere. Everything has changed. It changes all the associative organisms that the plant associates with also. And this gets me back to my point about the importance of mycorrhizae as a keystone species. The mycorrhiza association makes a glue that is used to glue soil aggregates together. And I know that having worked for NRCS, one of the things that we gave a whole lot of lip service to is soil health. And so all of the stuff that glues those soil particles together are basically mycorrhizal. So the glue is called glomalin. And this is interesting because I got into excuse me, I always get labeled as somebody that blames everything on mycorrhizae. And I had a colleague who was on the UAF staff named Jeff Smink, and Jeff was always giving me a hard time about soil aggregation and mycorrhizae. And I asked him what glue soil particles together, and he said, "Well, it's glomalin." And I said, "Glomalin is a name for the largest genus of mycorrhizal fungus. So literally that glue is named after mycorrhizal fungi. And so that glue increases soil aggregation and also carbon sequestration, which is another thing that NRCS is really into. So that keeps those aggregates keep carbon in the soil instead of in the atmosphere. So the glomalin produced by the mycorrhizal association can account for as much as 27% of the soil carbon out in the soil. On the lower left-hand side, you can see glomalin. This is an epifluorescence where they under a microscope, you bombard uh, the roots with a certain wavelength, and they'll turn around and autofluoresce. And you can see the spores down below that root. And on the right-hand side, you can see the glomalin that's gluing soil aggregates together. So benefits, mycorrhizae, they drive the evolution of plant adaptation to drought. One of the things that I was looking at in as in my PhD work, my PhD research, and my postdoc was how do different ecotypes of mycorrhizal fungi allow a plant to undergo water stress and still keep its stomates open, still keep fixing carbon. They continue to photosynthesize, and that's different based on the different types of or ecotypes of mycorrhizal fungi. Are you guys still with me? Hello? Well, I started to get into this plant adaptation to drought and the ecotypic, because it always, I was always working on like St. Helens when Weyerhaeuser they came out and put 150,000 Doug fir that they planted on Mount St. Helens, and those particular mycorrhizal fungi that occur on Doug fir are that association is fostered in a greenhouse in Federal Way, Washington, because they can inject that particular mycorrhizal fungus through the watering system. Well, somebody forgot to do it. And so all 150,000 Doug fir died from drought. None of them survived on Mount St. Helens. So, like I said, mycorrhizal fungi, they protect plants from drought conditions. They protect plants from pests by direct antagonism, like with the ectomycorrhizal fungi. Ecto just means outside. And those particular fungi, they produce compounds that will attack pathogenic fungi and pathogenic bacteria in the soil. And with endomycorrhizal fungi, which are the mycorrhizae that occur inside the host plant, they allow the plant to tolerate the pests like root knot nematode. You can get root knot nematode, and if the plant is mycorrhizal, you can have the nematode, but you really don't get a big reduction in plant growth. Another benefit of mycorrhizal fungi is an increased uptake of nutrients like nitrogen. Of course, phosphorus is the big one that everybody knows about, and micronutrients. There's also a tolerance to heavy metals, and I threw a, a citation down there below that. And I've seen mycorrhizal fungi on gold mines that allow big sagebrush to withstand. Whenever you have gold deposits, you usually have arsenic. And these particular mycorrhizal fungi were very tolerant of arsenic and allowed the big sagebrush to grow really well. And where you didn't have mycorrhizae, the sagebrush wouldn't grow. Soil aggregation is vastly improved, and down our I just threw in this big drag line from a coal mine I worked on, and I was working on jump starting plant succession after the coal companies came in and took all the coal out. And down on the right-hand side was a water stable aggregation study I did in Palmer, where I looked at whether or not plants were mycorrhizal. And you can see that it actually doubled the amount of soil aggregation in the soil. Mycorrhizal fungi enhance the rhizosphere organisms. I mean, so much so that a mycorrhizal plant, you cannot call it a rhizosphere, you have to call it a mycorrhizosphere. And I published a paper I put that on there, the variation in response of nodulating pigeon pea to different isolates of endomycorrhizal fungi. And pigeon pea is not a big plant in the United States, but it's one of the top five. Um, what am I looking at? Like lentils. It's like lentils. It's a nitrogen fixing pea. It's a legume, and it's very important in places like India. And so on the right-hand side, you can see I have two different ecotypes of the same particular mycorrhizal species. The only difference is one I isolated from Utah and one I isolated from California. The one from California just looks really puny and white, what you call etiolated. And that's because I found out that it's because what the mycorrhizal fungus did for the rhizobial bacteria that fix nitrogen. All the other organisms were basically the same. Mycorrhizal fungi also enhance these other microorganisms that mineralize organic phosphorus. And they can have them within the aggregates and also these bacteria that are phosphate solubilizing bacteria. They exist along the hyphae, which is basically a fungal term for root, kind of like a root. Just imagine fungus root. That's a hypha, and hyphae are a bunch of fungus roots. So these other organisms live along the root system. So the disadvantages, let's look at a disadvantage. The first one I can think of is operator error. And I'll get into this because this is the one thing I want people to realize is that you're dealing with a living organism. You are no longer dealing, mycorrhizal fungi are not a widget. You can't treat them, you can't plug them into a current cultural regime without altering your cultural regime. Because like I said, they've been evolving for 460 million years. They're not going to change overnight because you want them to. Like I said, you can't plug mycorrhizal fungi into your operation without acknowledging that you will have to alter what you're doing. Mycorrhizal fungi can actually compete with the host plant for nitrogen, and the mycorrhizal fungi out in the soil are better at pulling up nitrogen than the plant is. So that's another thing to keep in mind. And when you've got what I call easy phosphorus, which is very soluble phosphorus, and when you're adding soluble phosphorus like triammonium phosphate or DAP, um, diammonium phosphate, those really easy, what I call easy, what you're doing is making a dumb plant, a plant that's very used to having everything spoon fed to it. So under easy phosphorus, these soluble chemical phosphoruses, the mycorrhizal association can actually reduce the yield. And that's because they really shine when the phosphorus is hard to get at. And that really, I mean, you can always say, well, we're always going to have easy phosphorus, but phosphorus reserves worldwide, we're going through them pretty fast. And I don't know that it's my opinion that instead of, um, we're going to have a geopolitical problem in the mid 2000 by 2050 with regard to phosphorus reserves, because there's only a few countries where we get these easy phosphoruses from. We have to realize that in nature, no organism acts in a vacuum. So they throw one mycorrhizal species in there, and that's it. And they don't realize that in nature, that organism does not occur by itself. There's all kinds of other organisms. So which plants respond and which don't? It's a lot easier to say who is not mycorrhizal in nature than it is to list who is. Okay. So between 90 and 95% of our land plants enter into this mycorrhizal association with some form of fungi. Who has been assumed to not form mycorrhizae? And these are the plants. And I put assumed in quotations for a reason, and I'll come back to that. But, excuse me, unfortunately for Alaska, the Brassicaceae, or the, um, oh shoot, what did they used to call brassicas? I can't remember. But they're not mycorrhizal. So broccoli, cabbage, cauliflower, all these are thought to be not mycorrhizal. Okay, sugar beets, turnips, the mustard family, rapeseed, spinach. They're assumed not to have these associations. Anybody want to hazard a guess as to why?
Well, they take a lot of nitrogen, right?
What's that?
They take a lot of nitrogen, at least the cruciferae, the brassicaceae. I'm not sure if that's a relationship.
Well, one of the reasons, if you guys are familiar with World War One, when we had gas bombs, people were gassing other people to kill them, and the brassicaceae, like broccoli, cabbage, and cauliflower, they produce a compound in their roots that's called isocyanate. And isocyanate is a precursor to mustard gas. It's broken down into mustard gas, so it's actually poison for a lot of organisms. And mycorrhizae happens to be one of those organisms. But I said assumed because I did some digging and they've now found that the vesicular arbuscular mycorrhizae are in the Chenopodiaceae and the cruciferae. So you can tell those are old names because cruciferae is now called the Brassicaceae. So they have seen mycorrhizal fungi in the goosefoot family, which is the Chenopodiaceae, and in the family that broccoli, cabbage, and cauliflower are in. So you can't say anything for sure anymore. Turns out, yeah, and it happened when when the broccoli was grown with a known host plant. So the mycorrhizae get in and around. If you put nothing but broccoli out there, you probably won't get colonization. But if you intercrop with a known mycorrhizal host, you may wind up getting colonization in your broccoli. Okay, I had to throw this page in. I had a picture of of a deer caught in the headlights. Um, this is a science page, and I hate to bring it out, but it's instrumental in what happens from here on out. So a symbiosis is just a relationship between organisms of different species. Okay, and there can be a mutualistic symbiosis, which is where both organisms benefit, and that's what the two pluses are. There can also be a parasitic symbiosis where one organism thrives at the expense of the other. And there are some other symbiotic associations, but I didn't want to get into those because mutualism and parasitism is more than enough to remember right now. So if you have the mutualistic symbiosis where both organisms are benefiting, you can have what is called an obligate mutualistic symbiosis. And I tried to figure out a way to write that in everyday English, and I came up with the fungi say, I just can't live without you. Okay, so the fungi, these obligate symbionts or obligate symbiotic organisms, they can't live, they cannot complete their life cycle in the absence of a host plant. Okay, facultative mutualistic symbiosis plants, or associations, are where the fungus says, I prefer our relationship, but I can make it without you, without the host plant. Okay? And that's pretty imperative because that totally changes the way you have to treat these organisms, how you have to grow them, to bulk them up. And this is one of the biggest errors I've seen people do with mycorrhizal fungi. I've seen it down on the Kenai Peninsula. I've seen it just about everywhere. There's a lot of misinformation. Okay. So an obligate mycorrhizal association or obligate mycorrhizal fungus needs an actively growing root. So this is an on-farm inoculum production with bags showing late summer abundant growth of Bahia grass. And Bahia grass is, is a grass that produces a lot of sugar. Okay, so the take-home point is obligate mycorrhizal fungi need an actively growing root. Therein lies the problem with ProMix. Does anybody else see that? Because ProMix, they don't have a very high count, like 5 propagules per gram of soil, which is nothing to write home to mom about. But they're actually attempting to say that those fungi will grow in that block of peat moss, and it won't happen because they cannot grow in the absence of a growing root. And so these Bahia grass are in these bags, and that's where the fungi are down inside the bag, and they're growing that way. Okay? A facultative mycorrhizal association does not need actively growing roots. You can increase their numbers in a laboratory. And I've done that where you inoculate broth and you spin it and they'll grow perfect. And this gets me to another point, and we'll get back to that. But the point is people saying that mycorrhizal fungi can be increased in compost tea. That is a myth, depending on which mycorrhiza you're talking about. Okay, this is another take-home point. If you don't take anything else out of this classroom or off of this presentation, take this home. There are 7 different types of mycorrhizal associations that associate with 7 different types of plants. Okay? The ectomycorrhizae. Ecto just means outside. So the main vegetative portion of the mycorrhizal fungus is outside of the plant. So these are I prefer being with the plant, but I can make it on my own. So that means ectomycorrhizal fungi are perfectly happy living in the soil by acting like saprophytes, which just means death eater. So they are breaking down carbon sources in the soil. They prefer to be with a Doug fir or a spruce, but they can make it on their own and wait until there's a host that comes along. The endomycorrhizae are different from the ectomycorrhizae in that their main vegetative portion are inside the host root, and they are the I can't live without the plant. But some are I prefer the plant, but I can make it without the plant. So it doesn't all nothing is always there's no blanket statements. Okay. So I put this in, and on the right hand side, you can see the ectomycorrhizal fungi, and you can see the red tangled up hyphae. So the hyphae, unlike, when I said about the rigid cell wall and the cell membrane being like a latex glove, you don't have to worry about that with these because they will not penetrate the cell wall. They'll grow in between the root cells, but not into the root cells. Okay? The far more common type are the endomycorrhizae, and the hyphae will penetrate the root cells. And you saw that first slide I had with that tree-like structure, that was inside of a root cell. So I'm going to elaborate a little bit on endomycorrhizae. So I'm going to elaborate on the I can't make it without you fungi, the different types. So there's orchid mycorrhizae, which is I can't make it without you. And they occur in all 35,000 species of orchid. There's the arbuscular, in other words, the tree-like looking mycorrhizal fungi. They're also, I can't make it without you. And they're in about 220,000 species of plants, including most of our food crops. There's the ericaceous mycorrhizae. And the ericaceous mycorrhizae are the I prefer you, but I can live without you. And they occur in the blueberries, the vaccinium, all the vaccinia genera, excuse me. The arbutoid mycorrhizae, they're in bearberries and pyrola. This picture down here is a pyrola acerifolia, which is a wintergreen. It's in the woods, but that's an arbutoid mycorrhizal fungus. And then there's the monotropoid mycorrhizae, which occurs in Indian pipe. And you can see that Indian pipe is a plant, but it doesn't have any chlorophyll. It's not green, it's white. How can it do that? It forms an association with a tree below it, and it gets its nutrition across those mycorrhizal hyphae that attach the tree to the Indian pipe. Okay, I'm going to switch gears a little bit. This is where I can thank Rita Joe. I gave this talk in 2013 or a talk like this in 2013 in Fairbanks. And I went out to dinner afterwards, and Rita Joe went out to dinner with me and some other people, and I asked Rita Joe, I said, Rita Joe, what'd you think of my talk? And Rita Joe looked at me as Rita Joe will often do. She shoots straight from the hip. She said, I liked it, but just tell me what I have to do. And so I thought, okay, I'm going to go home and I'm going to write up and I'm going to get a flow chart so I can answer Rita Joe's questions. And I sent her this flow chart about 11 years ago. So, first question, is my plant mycorrhizal? What is your goal? Should I make my plant mycorrhizal in your operation? What is your goal first? Is it strictly nutritional? You just want to increase your plant's nutrition? Or are you looking to sustain your soil health? And then you have to ask the next question you have to ask yourself, if it's strictly nutritional, do I have easy soil phosphorus levels in my soil? And when you get a soil sample back, it is the available soil phosphorus. That's not the total soil phosphorus. It's what's available to the plant. And so first question I say is it less than 70 parts per million or less than 140 pounds per acre of easy soil phosphorus? And then you have to ask yourself, the next question is, what is my plant? If it's greater, if your easy soil phosphorus is greater than 70 parts per million on 140 pounds per acre, then you have a whole different problem. You may not get any colonization at all. The plant, when plants are starved for phosphorus, they send signals out into the soil, and these signals, here's a $10 word, are called strigolactones. And these strigolactones, they serve as a stimulus for mycorrhizal spores to germinate and grow towards the source of that strigolactone, which happens to be the plant. And it's because the plant, when they're starved for phosphorus, their cortex, their root becomes leaky and leaks these strigolactones into the soil. Your second question has to be, okay, my plant is my plant, is it a gymnosperm? Gymnosperm just means naked seed. I don't know if you've ever pulled a pine cone apart, but those seeds are not held within a fruit. They are naked. Again, they just have what's called a pappus or a wing. So those are the pines, the cedars, spruces, firs, oaks, some of the aspens that we have. So it's those plants are gymnosperms, and those are ectomycorrhizal. They so if you inoculate with an endomycorrhizal fungus, you inoculate your pine with it, you might as well you just you might as well spit into a stiff breeze because you're not going to get any colonization. You're not going to get any of the benefits. If you are growing Doug fir like they did for St. Helens, yeah, the ectomycorrhizal fungi are perfect. And I have a picture, you can actually see four supercell pines, and the one that's second from the right has got the best example of ectomycorrhizal fungi in it. So what happens if my plant is an angiosperm? Okay, angiosperm just means that the seed is born within a fruit. A tomato is an angiosperm. Everything that we have that's born in a fruit is an angiosperm. So there's, like I said, there's about 220,000 species of angiosperms that are mycorrhizal, and there's about 10,000 species of plants that are ectomycorrhizal. So if your plant's an angiosperm, like um, Rita Joe's peonies, they're an angiosperm. They are not a pine, they're not a cedar, they're not a spruce, they're not a fir, they're not an oak, they are an angiosperm. So that sends Rita Joe down this branch of the flow chart. So the next question you have to ask yourself, if they're endomycorrhizal, are, are they blueberries? Blueberries are endomycorrhizal, but they're endomycorrhizal with with ericaceous mycorrhizal fungi that will only grow with blueberries and cranberries. And that's where you have to use an ericoid mycorrhiza. Okay, that's what you're trying to foster. So you see all these questions that you have to ask yourself when you're trying to grow stuff. Uh, is your plant not a peony, but is it an orchid? Okay, now you're talking about a different mycorrhizal fungus. You're talking about an orchid mycorrhizal fungus. Is it a brassica? In which case, you're going to assume that it's non-mycorrhizal unless you feel like doing a lot of research and figuring out whether or not it may actually have mycorrhizal fungi or not. And everything else, other than ericaceous, orchid, brassica, gymnosperms, everything else is an AB mycorrhizal fungus. That's the arbuscular mycorrhizal fungus. Next question you have to ask yourself, where do I put these fungi in order to get the best bang for my buck? Okay, so you've got two questions to ask here. We're back to, is it an AB or an endomycorrhizal, or excuse me, is it an AB mycorrhiza or is it an ectomycorrhiza? So let's say, I split this out because mycorrhizal inoculum used to be really expensive back 30 years ago. It's not so bad now, but you still want to maximize your cultural regime in order to get the best colonization for what you're doing. One of the two questions that you have to ask is, what is the spore size? Because ectomycorrhizal fungi are really small spores. They will percolate down through a soil column. Endomycorrhizae, the AB mycorrhizal fungi especially, are huge by comparison, and you'd have as much success attempting to shove a basketball into the soil. They're just huge. Some of them you can actually see with the naked eye without a microscope. They're just very big. If you are growing, let's say you are growing firs, somebody asked you to grow Doug firs to plant somewhere, you have to realize that these ectomycorrhizal fungi are extremely host specific. Whereas the AB mycorrhizal fungi are what they call a promiscuous fungus. In other words, one fungus will colonize with 1,000 species of plants. And then once you get that question answered, you should, in order to maximize your colonization, you should look to see whether or not is my plant taprooted or is it fibrous? Am I growing wheat or am I growing a tomato? Tomatoes are rather taprooted species. Um, Another one would be alfalfa. But unfortunately, alfalfa doesn't grow very well in Alaska unless it's Denali. So if you are growing, you have to ask yourself these questions about the root to figure out where you should put it. Should you should you concentrate your mycorrhizal fungi in a horizontal band when you've got a fibrous root system that's going to branch out quite a bit? Or should you concentrate your fungal inoculum in a vertical column? And I did that. That's one of the things I did in my PhD was where to concentrate AB mycorrhizal fungi with Western red cedar. Um, all right. So you have to know where to put it. You have to know how much to put on. You have to know which fungus you're dealing with. You have to know which plant you're dealing with. And then you have to understand, like I said, when should I put them on? With the AB mycorrhizal fungi, you use less if you know where to put them and when to put them. And you put them on as early as possible when the plant is in the most controlled environment you can achieve. With the ectomycorrhizal fungi, you put them on as early as possible, also in a controlled environment. Unfortunately, like I said, for Weyerhaeuser, somebody forgot to do that in the greenhouse. One of the things, the take-home message here is you can deliver the ectomycorrhizal, they'll water in. You can deliver them to a plant that's already growing. The AB mycorrhiza, it behooves you, it's more advantageous for you to plant your seed above your mycorrhizal inoculum so that the root has to grow through it. So with one, you can deliver the spore to the root. With the other, you have to deliver the root to the spore. And in both cases, you're going to have to alter what you're doing. You're going to have to cut way back on your easy phosphorus. Or you can take advantage of what I call the kitchen sink. This is, what the heck is Down to Earth? I think this is Down to Earth. And I took a photograph of their bag. And here you can see they've thrown 1, 2, 3, 4, 5, 6, 7, 8, 9 different species of AB mycorrhiza, which we know will work with tomatoes. And they also threw in the same bag 1, 2, 3, 4, 5, 6, 7 ectomycorrhizal fungi that won't work with tomatoes. I mean, you might as well throw those out. But anyway, they're covering, I call it the CYA, which is cover your hiney. Okay. They also threw in some biocontrol organisms like this trichoderma, which will attack pathogenic fungi. Excuse me. And they threw in bacteria, which are PGPRs, which are plant growth promoting rhizobacteria. And those are going to help the mycorrhizal fungi to colonize. Okay. You guys still with me? Am I starting to bore you? Okay. I think this is it. We're almost there. And here it is, my big spiel. The mycorrhizal conundrum, or why can't I grow that myco thing in my compost tea? Mr. or Ms. I'm a farmer wants to grow their own mycorrhizal fungi to inoculate their tomatoes and have heard they could do it in compost tea. They get some AB or endomycorrhizal fungi and separate out 50 spores or propagules. They dump them in their compost tea and they start that tea a bubbling. The farmers keep a keen eye on their AB fungi and strain out what they think will be a whole lot more fungi 6 weeks later. They think, hurrah, my compost tea increased my mycorrhizal fungi. They start counting and they notice nothing. Their propagules, their spores ain't done nothing. And in fact, a lot of them died. And does farmers think, what did I do or what did not happen? Anybody have an idea? The farmers, they need to understand the difference between the 7 different types of mycorrhizal fungi and how they have evolved to grow the way that they do. The mycorrhizal fungi that will help their tomatoes cannot reproduce without a living plant. They need a living root exudate. No living plant inside the compost bucket equals no increasing the fungal seeds. Okay? The mycorrhizal fungi that will potentially grow in their compost bucket may help with white spruce on their property, but won't do anything for their tomatoes. These ectomycorrhizal fungi, remember, these are the I prefer you, but I can live without you fungi. The ectomycorrhizal fungi do fine without a living plant as long as they have some source of carbon to break down. So no living plant inside the bucket equals no problem increasing your fungi. And I threw in an AB mycorrhizal picture on top and an ectomycorrhizal picture on the bottom. And I want to state, at least to kind of show you what ectomycorrhizal fungi look like, these are 3 pine seedlings. And so the pine root is, there are 3 pine roots growing from that middle pine seedling. Everything else that you see is ectomycorrhizal hyphae. And mycorrhizal fungi, they can increase the volume of soil that a plant can mine for nutrients and water by up to 10,000 times. So I tell kids, when I'm giving this talk, I say, hold out your hand. Now open up your hand. Your fingers are the volume of soil that that your roots can get nutrition from. Now imagine that your hand is 10,000 times bigger. That's what happens when you inoculate with mycorrhizal fungi. So in the future, we just need to, somewhere between Sarah Wright, who's at Beltsville Area Research Center, who discovered glomalin as a glue, and I'm a farmer, we need to clear up some of this misinformation, because otherwise you're not using the mycorrhizal fungi properly. It won't work, and then you'll blame the fungi instead of the way you did it. And I just threw in these because there's, on the left-hand side is an ectomycorrhizal fruit. The tree, and I want to emphasize this, the tree that fruit is coming off of is below ground. That's the part you can't see. And I threw in Western red cedar on the right-hand side to show you what happens with Western red cedar, because it actually did put in a fibrous root system. And so when I banded the mycorrhizae, I get this huge growth spurt. And I will entertain questions.
Well, great.
Are you guys thoroughly confused?
Okay, we'll get our camera back on so you can see us while we ask questions.
Okay.
Folks on the Zoom, you can use the chat box, or you can always unmute yourself and go ahead and ask live, whichever you prefer. We'll start with the room to give the Zoom folks some time, so.
Hi, Steve.
Anybody here got an opening question for Mr. Ianson? Yes, go for it.
Working with mycorrhizae, have you taken into account or noticed a specific bandwidth between mycorrhizae function and temperature?
Once they've colonized the plant, or just in general?
Their performance in general, if they have a specific temperature range they function best at, or when does it become to be seriously detrimental or kill mycorrhizae?
Well, okay, I'm glad you asked that, because I'm glad that you elaborated, because when you when you solarize soil or compost, you can, if the goal is to increase the soil temperature up to what is required to kill pathogenic organisms, you will have long since killed the mycorrhizal spores. So fungi, you get up above about 110 degrees or 105 degrees, you're going to start to kill the fungal spores off. Does that answer your question?
What would you consider the low temperature range before you start to see mycorrhizal death?
Well, they'll go quiescent. They'll go into a resting state and they'll withstand, freezing. So you can
Perfect.
so once you get below ground, because the whole system is below ground. So the soil surface and the soil acts like a huge buffering system against rapid changes in soil temperature.
What about activity level at different temperatures? I had someone come through my farm last year who said my soil fungi were sleepy. And I wondered if it had to do with soil temperature. It was,
Did
Pretty
they
cold.
elaborate on the term sleepy?
it did not.
Okay. That's kind of how high is up.
Yeah, yeah. But is there, I mean, I guess the question is, are we at a disadvantage in Alaska because our soils are so cold?
We are at a bit of a disadvantage from the term of the AB fungi that, like, people that want to grow certain plants outdoors without a high tunnel. And but the ectomycorrhizal fungi, you go out, you look at the white spruce in your property, they're going to be loaded. And I've pulled out endomycorrhizal fungi from areas, native areas around agricultural fields. So they are there. You get up above, I mean, where you guys are, it's probably not a problem. If you get up above maybe the Brooks Range, and I've pulled mycorrhizal fungi out of everywhere except Antarctica. That's about the only place I've not. I pulled them out of soil from Tierra del Fuego on the very bottom of the South American tip. And I pulled them out of soil up here and you actually used mycorrhizal fungi to jumpstart um, uh, what's that bush called? Well, up in Nolan, I worked with jump-starting succession on a gold mine up in Nolan, and I used mycorrhizal fungi, and they had no problem.
All right, well, let's take a couple of questions out of the chat. I've heard that trichoderma will attack beneficial fungi as well. Have you seen it be detrimental in mycorrhizal fungi?
Not yet, but that doesn't mean it doesn't happen. But you did notice that they will put that Down to Earth did put Trichoderma harzianum in the same mix with endos and ectos. So I doubt whether the people at Down to Earth would put something that would attack something else in their mix, unless they're really trying to pull one over on us.
They're really do.
All right, well, related to the trichoderma again, our trichoderma or Bacillus subtilis,
Bacillus?
it's in the chat box. Are they appropriate for fertigation in peonies? And this person has had you help them a little bit with their peony fields before, with a variety of inoculants for their cover crops, Austrian field peas, buckwheat, clover, prior to planting. The fields are quite productive and healthy, but they're wondering if they need to do anything to maintain the mycorrhizal fungi levels in the fields, or once inoculated, can you just let it go?
Well, rotate with a host every now and again with a cover crop that's a host. Um, I mean, some of the grasses, wheat, um, other legumes work well. Um, I've turned around and used Austrian pea as a cover crop, and they'll foster mycorrhizal fungi really well. So the fungi, if a host plant is not there, the AB fungi will go dormant and quiescent and just kind of they'll get kind of a hard spore shell and just kind of wait for a period of time. The ectomycorrhizal fungi that occur on the firs and not our crop plants, they will turn around and start breaking down dead organic material until another host comes along. Does that answer the question? Keep something, keep some kind of a host rotating through the system.
So David, she grows peony rootstock, which is a perennial.
Right.
At that point, she would not be able to probably do cover crops.
Oh, okay.
So
Well,
I think
they'll
fertigation question, can she use something through her fertigation system when she feeds her plants?
okay, I get what you're saying now. The problem, the potential problem with that is we are one, assuming that her peony roots are already established, correct?
Correct.
Okay, if she fertigates on the soil surface and the propagules or the trying to find a word that makes sense for folks, but a mycorrhizal propagule can be a spore, it can be a hypha, or it can be a piece of the previous host. The problem with anything but the hypha is that the mycorrhizal spores, some of them can be really big. If, if they are fertigating on the soil surface and the soil is kind of a clayey rather than what I would call a lighter soil like a sandy or a sandy loam, then those endomycorrhizal propagules will not percolate down through the soil. Having said that, what I would do, have you ever seen one of those? I've actually got one. It looks like a, looks like what do you call them? A meat injector, that you inject different types of thing into your roast. They make one of those that's big enough to use in your yard. It's how to get water down to your root system. So you can, what I have done in an established plant is gone in with that thing, and it has holes big enough to where you can inject the mycorrhizal fungi in your watering system down into where the roots are.
Okay.
Like I say, the problem with anything other than perhaps the hyphae can percolate. They're really small, but the spores can be really big, as can pieces of the previous host.
All right, so that brings us to a very related question also in the chat. How are you applying the mycorrhizae in the field, or how do we go about applying them in our garden? So you've mentioned the the meat injector method. What are some of the other ways?
Well, I've worked with them on strawberries, and you can buy inoculum like that. I showed you that bag from Down to Earth. You can buy that inoculum, and they make inoculum or inocula that you can put in water. And with strawberries, I've mixed up that inoculum, and it produces this soupy dark brown stuff that's all spores and whatnot. And then you just drag the roots through there and plant the strawberry roots into the hole. Um, with other plants, you could do it that way also. It gets to be cost prohibitive when you're trying to broadcast mycorrhizal inoculum or inocula across the entire field. I think you're almost better trying to foster any fungi that are already there. The return's a lot better with peonies than it is with somebody trying to grow alfalfa. Let's put it that way. Does that
Any
make
questions?
sense? Does that make sense? It depends on your return. High return crops, you can afford to use more inoculum. Low return crops, you have to start asking yourself, what are the cost benefit ratio?
Can I ask a
In
question?
a garden, I could always pencil that out. But in a
So-
field, that's a different animal.
So I know a few farmers that I follow who do no-till, and they talk a lot about inoculating all their vegetables, perhaps not their kales and cabbages and stuff, with mycorrhizal fungi,
Fungi,
fungi. I just
they're
can't do it. Gee,
they're
it's okay.
they're
Um,
inoculating with mycorrhizal fungi in hopes of getting a mycorrhizae.
Yes.
Yeah.
Exactly. But I thought I heard you say at the beginning of the conversation that these ones for vegetables are not water soluble. So is that actually worthwhile doing? Because there's lots of products out there
Oh,
and
they're water soluble. They just don't, if you put them on the soil surface and try to water them in, you're not going to have as much success.
okay.
You have to, with that particular fungus, you have to deliver the root to the fungus, not the fungus to the root. Does that make
So if you,
sense?
yeah, so if you had a tray and you were dipping your starts, in the tray, like say they're in a cell pack or something, and so you're saturating the soil from the bottom up with that inoculant, that could work.
Well, you would be dependent upon a wicking action. And if you had a light soil, you're not going to get as much wicking as you would with a more loamy or a clayey soil. Um, you would be dependent upon that. What I would do on a supercell, or a tray pack, is when I put them in the ground, if I had a machine, I had a transplanter, as as the as the supercell came around, I might squirt a little bit of inoculum down in the hole and put the plant in on top of it.
That sounds like a lot of work.
It is a lot of work. I know it is a lot of work. Yeah. You know what I did? I did a cover crop study here in Palmer where I used endomycorrhizal fungi, and I just mixed it in. I mixed the inoculum in with the seed and just put them down all together. I just dipped my hand in the bag with the inoculum and the seed and threw it out like I was feeding chickens.
And did it colonize?
Yeah, yeah. I had to come through with a rake, and I call it scratching. I don't want to say tilling, because tilling is a death knell to the mycorrhizal fungi, especially the hyphae. You just break up all the hyphae. But I came along with a great big landscape rake, and I just dragged it across the surface afterwards.
All
The
right,
only
well,
the only problem that I had was the farmer whose field I put that in, he already had like 470 parts per million phosphorus. I still got colonization, but Jeff and I were arguing about, because he said, well, the non-mycorrhizal ones eventually caught up. And I said, yeah, if you're putting 400 or 840 pounds per acre of phosphorus, but who's got that much phosphorus?
Okay, so that kind of brings us to a chat question and then one that I have that goes right in tandem with it. So you're distinguishing between the fungi that are already present in the soil. I mean, there's fungi everywhere, right?
Correct.
And then the specialized kind of mycorrhizal fungi. So isn't there mycorrhizal fungi already there of some variety? This is just, you're inoculating with a more potent kind, or what makes?
Well, you are concentrating, so you may wind up like, let's get back to the pro mix thing. Your pro mix says 5 propagules per gram of soil. In that Western red cedar experiment I did, I used 1,000 propagules per gram of soil. So what you're doing is increasing your chances quite a bit.
So that brings it to the chat question. How do you were talking about, you may as well foster the fungi that are already there.
Right.
If you're given some certain circumstances, how do you do that?
Don't till for one thing. Keep the soil covered for another. That gets us back to the heat thing. You got an organism that's developed in the dark and in cool conditions, and all of a sudden you're leaving a field fallow and the sun's hitting it, and it's just cooking the entire surface. Um, so that's another thing. I'd keep it covered, keep a host going through there if I can. Um, just stuff like that. Try to foster soil health, and the fungi will come along. It may take them a while because the ectomycorrhizal fungi they can get in the gulp in the whatchamacallit stream, the up in the air, and they can blow for thousands of miles. But the AB fungal spores, they're so big. I did a study in Wyoming and I got them. I was picking them up at a mile, but then again, Wyoming blows like crazy too. Anything that transports soil is going to transport AB mycorrhizal propagules.
Hey, is it all right if I ask one of the questions from the chat? Is there a recommended soil test to check for mycorrhizia? I'm gonna see if I can get it right. Nah, can't do
Fungi.
it, John, I just can't do it, I'm sorry.
Mycorrhizal fungi.
I'm gonna practice. I should have practiced before, but is there a soil test?
There is. I used to work with a lady in Oregon in Corvallis by name of Elaine Ingham, and Elaine Ingham has a company called Earth Fort, and she may have a mycorrhizist on staff. I used to be the mycorrhizist for her, but that was a long time ago. But anyway, she may have somebody doing counts. The other way of doing mycorrhizae is kind of throwing it in with all the other organisms, and they have what they call the Solvita method, where they're measuring respiration of the soil and kind of calculating that back to all the microorganisms. That doesn't particularly break out any particular species of organism. It doesn't separate the fungi from the bacteria and that kind of stuff. I've spent a lot of years going through soil and sieving it out and just counting the number of spores in a gram of soil.
Can you see these under a microscope yourself? The AB?
Yeah, if you know what you're looking for. And if you know what you're looking for, there's a couple of species you can see with the naked eye. They're, like I said, they're huge. They're 1,000 microns across, where the ectomycorrhizal fungi are maybe 30, 37 microns across. So you can see why those ectomycorrhizal fungi get up into the jet stream and get blown all over the place. they're, what is that? 1/100th, 1/200th of the size of an AB spore.
We got a question about brands. If you're gonna try to buy give yourself better chances, get a big bunch of them. Do you have any recommendations on how people can buy that stuff?
To buy what?
Buy either brands of
Oh, okay.
places
Yep.
or places to look.
What I tell people, and it requires a little bit of background work on the part of the buyer, but I look for, number one, I look for a company on the West Coast, particularly the Northwest Coast, and I look for whether or not they have a mycorrhizist on staff, and that's where I buy stuff. There's an outfit that it was called Mycorrhizal Applications, and I sent a PDF to Monica this afternoon with a list of plants and mycorrhiza from Mycorrhizal Applications. But I know the guy I know the guy that used to run that particular operation, and he's a well-known mycorrhizist. Um, like I said, myself, I would look to see if there's a mycorrhizist on staff at a company. And there's an outfit called Fungi Perfecti out of Olympia, Washington, and that is run by a guy by the name of Paul Stamets, Stamets, Paul Stamets, and he's really knowledgeable.
David, I
Yeah.
have a mountain of chips, spruce chips with some hardwood mixed in, a year old, that you can see the hyphae in the pile, but it's dead. There's no living thing to feed them. Is that going to kill if I use some of Stamets spore to grow mushrooms in that substrate? Will that hyphae in there attack them, or does it need to be sterilized before you could inoculate it with your
Well,
mushroom?
one of the things you kind of have to remember is that fungi are divided by whether or not they're saprophytic. In other words, all they want to do is break down woody material or carbon sources. You have
Right.
other fungi like that trichoderma that will attack other fungi, and but then again, like I say, that outfit has put trichoderma in there, Trichoderma harzianum, and apparently it works, and it probably reduces the competition by other fungi on the mycorrhizal fungi, and then you have the beneficial fungi. So getting back to your point, if you've got a lot of hyphae in your wood chips, it's a good bet that they are saprophytic fungi, that they're living off the wood, in which case they will not attack the ectomycorrhizal fungi or the endomycorrhizal fungi.
And there's no visible way to tell whether they're parasitic or saprophytic?
Not until you give it a try.
Okay.
One of the things that, there's a guy, he's out of, where's he out of? Nikiski. He was giving a talk when I was down in Kenai with NRCS, and he was talking about, he's a composter, and he said that in order to inoculate his compost, he went out to the woods and he got mushroom caps and he put them all over the compost. And my first question was, what are you trying to grow? And he said crops. And I said, well, if your crop is spruce, I can see where it would work, but not with what you're trying to grow. If you're trying to grow, all these other things like tomatoes and potatoes and all the other 220,000 species, no. Putting mushroom caps, because mushroom caps, like the last picture I showed, that Amanita, that is the fruiting body of an ectomycorrhizal fungus. If I cut that cap off, put it on a compost pile, and try to inoculate tomatoes, it's just not going to work. And that's kind of the what I want to leave you guys with is they're not all created the same and they don't all work the same.
We've just got time for maybe 1, 2 more questions because we've gone over and want to respect Dave's time. In the chat box, Justin Whitaker has mentioned that he has opened a lab using Dr. Elaine Ingram's techniques taught at her school. He doesn't do root standing or mycorrhizal, just total fungi. That business is called arcticrootsanalytics.com, and I know he's out of Esther, I believe, Alaska. And he comments about Earth Fort no longer being a regional lab, but that techs have been trained there to continue that work. Do we have any more questions for Dave Ianson from inside the room? No, everybody's satisfied for right now with the thing you just left us with. Chat box folks, are you guys good to go?
Remember, with your endomycorrhizae, you're going to deliver the root to the fungus. If you should happen to be working with ectos, which I can't picture unless you're in the lumber industry, then you can deliver the fungi to the root. One is the root to the fungi, the other one is the fungi to the root.
Right, well, thank you so much for your wisdom, Dave, and all your years of studying this completely fascinating topic.